The topic selected for today are osmosis, plasmolysis and imbibition. You know that pure water has highest water potential, and as the concentration of the fluids increase, the water potential correspondingly decreases. This point has great bearing on osmosis. Now let us see what is osmosis. Osmosis is defined as passage of water molecules from a region of higher concentration, that is, from a region of higher water potential to a region of lower concentration, that is, the region of lower water potential, till an equilibrium is established. So, if two solutions with a different water potential are separated by a semi-permeable membrane, water molecules always move from a region of higher water potential to a region of lower water potential. Now I have drawn here a figure where two chambers A and B are separated by a semi-permeable membrane. Chamber A is filled with solution having higher water potential, that means less diluted, where chamber B is filled with solution with lower water potential, that means a concentrated solution. So diluted solution means it has higher water potential, and the concentrated solution means it has lower water potential. So here two solutions, diluted solution and the concentrated solution are separated by a semi-permeable membrane. Semi-permeable membrane allows water molecules to move freely across that membrane. And you know that water molecules have kinetic energy. Water molecules are moving always in different directions. So naturally, some water molecules from chamber A move into chamber B, and similarly, some of the water molecules from chamber B move into chamber A. But here, because of the difference in the concentration gradient, more water molecules move from chamber A to chamber B. That means the net movement of water molecules is from chamber A to chamber B till an equilibrium is established. That means till the concentration of the solutions present in chamber A and B become isotonic, the water molecules continue to pass through the semi-permeable membrane. And when an equilibrium is established between A and B, there will be no net gain of movement either by the chamber A or chamber B. So that is about osmosis. That can be demonstrated through a potato osmometer experiment. If you take a potato tuber and make a cavity and fill the cavity with sugar solution and place the potato in water, what happens? Because of water entering the potato, because of the difference in the concentration gradient, the solution, the sugar solution level in the cavity of the potato tuber increases. This clearly indicates that water moves from higher water potential to a region of lower water potential. Then, in your book, some questions are given based upon osmosis. Now let us find out answers for those questions. Solution of which chamber has a lower water potential? You know that water potential is highest in pure water. As the concentration of the solution increases, the water potential decreases. So here, chamber A has higher water potential and chamber B has lower water potential. So the correct answer is chamber B. The second question, solution of which chamber has a lower solute potential? As the concentration of the solute increases, it lowers the solute potential. So here chamber B has more solute molecules. So the correct answer is chamber B. Then the third question, in which direction osmosis occurs? So water always, or the net movement of water will be from a region of higher water potential to a region of lower water potential. Higher water potential is chamber A and lower water potential is in chamber B. So naturally, the net movement of water will be from chamber A to chamber B. Fourth question. Which solution has a higher solute potential? Again, as the fewer the solute particles, the greater will be the solute potential. So here, chamber A has fewer solute molecules or particles. So chamber A has a higher solute potential. Then, fifth question. At equilibrium, which chamber will have lower water potential? So when an equilibrium is established, there will be no difference in the water potential of either chamber A or chamber B, because the osmotic pressure or the water potential is the same in both the chambers. So the answer is both the chambers have equal water potential. The last question. If one chamber has C means water potential of minus 2000 kilopascals and the other has minus 1000 kilopascals, which is the chamber that has highest water potential? So here, chamber A, let us say it has minus 1000 kilopascals and chamber B has a pressure of minus 2000 kilopascals. That means chamber A has highest water potential and chamber B has lowest water potential. That's why the answer is chamber A, that has 1000 or minus 1000 kilopascals. That is about the answers for the 6 questions set in the textbook. Then coming to the structure of a plant cell. You know that plant cells are surrounded by a cell wall. Cell wall is porous in nature and it is freely permeable to water and solute molecules or solute particles. So it is not a barrier for the movement of water and solutes. Then the cytoplasm is surrounded by a plasma membrane. In the cytoplasm we find a nucleus and also a central vacuole. The central vacuole is filled with cell sap and surrounded by a membrane known as tonoplast. In the cell sap we find a number of dissolved sugars, salts, etc. The main difference is the cell wall is not selectively permeable, whereas that means selectively permeable means it is freely permeable, where the plasma membrane and the tonoplast are selectively permeable. That means the cell membrane and the tonoplast choose which substances to be allowed inside and which substances to be allowed to leave the cell. So they are of great importance in osmosis. Then, coming to the process of osmosis, solutions are 3 types. Hypertonic solutions, hypotonic solutions, and isotonic solutions. Hypertonic solutions have lesser water potential than the cytoplasm. That means the amount of water present in the solution will be less than the water present in the cells or in the cytoplasm and the cell sap. So what happens if you place a plant cell in a hypertonic solution? Here, this is the cell, this is the hypertonic solution. Then what happens because of the difference in the osmolarity? Water moves from the protoplast into the surrounding fluid. So the cell loses water as the water moves out of the cell, and the water moves till an equilibrium is established. Again, the water potential of the cell is here, the water potential of the cell is more than the water potential of the surrounding fluid. As more and more water leaves the cell, what happens? The protoplast shrinks in size and it is not in a position to exert any pressure on the cell membrane. That means the protoplast lacks any turgidity. It cannot exert any turgid pressure on the cell membrane and the protoplast shrinks in size. But this process is called plasmolysis. Loss of water from the protoplast into the surrounding fluid is known as plasmolysis. Plasmolysis can be reversible. If the plant cell is placed in pure water, what happens? Because of the again difference in the water potential, water moves from outside into the cell and the cell regains its original shape and its turgidity. So, plasmolysis is only a temporary process and it can be reversed. Then, hypotonic solution. Hypotonic solution means the solution has greater water potential than the cytoplasm. That means cytoplasm has solutions in the higher concentration, whereas the surrounding fluid has solution in a lesser concentration. As a result, what happens? Because of the difference in the water potential, water moves from outside into the cell. As a result, the turgor pressure inside the cell increases as more and more water diffuses from the hypotonic solution into the cell. The turgor pressure inside the cell increases, and this happens till an equilibrium is established. Then, isotonic solution. In the case of isotonic solution, the water potential of the cytoplasm and the fluid will be the same. If a cell is placed in isotonic solution, as there is no difference in the water potential of the cell and the surrounding fluid, no net gain happens either in the case of cell or in the case of the surrounding fluid. In this way, the cells behave differently in three types of solutions. Then, coming to the imbibition. It is a special type of diffusion where water is absorbed by solids and colloids, causing them to increase in enormous size. So, it is a process where some colloids and substances increase in great size by absorbing a lot of water. This type of behavior is seen in seeds and dry wood. You know that seeds have very little water, and when you place a seed in water, what happens? It absorbs water through micropyle, and the seed swells in size because of imbibition, absorption of more and more water. And this creates an enormous pressure inside the seed. As a result, the seed coat is ruptured and the cotyledons come out. And this imbibition force also helps the seedlings to emerge out of the soil. Then, in the olden days, the imbibition force was used by our ancestors to split rocks and boulders. If a dry wood is placed in a rock, in the cavities of a rock, what happens? And put water, the wood absorbs water and the imbibition pressure increases greatly. This increased imbibition force or pressure splits the rocks and boulders. In this way, in the olden days, the force of imbibition was used by our ancestors to split open the rocks and boulders. So that is about osmosis, plasmolysis and imbibition. Thank you.